спасибо. Да. Мгм. Так, а листаю я. Все, начинаю, да? Good morning, dear friends. Please indicate your presence. I have just seen first students very well, but why only one? Okay, we'll start consider today the next topic on physiology. It's physiology of respiratory system. Maybe we can say that respiratory system is the most important system because as it expressed in Latin proverb, dum spiro spero, that means until I breathe, I hope. From this proverb, we understand that, and we know that when this system stops its activity, it means death of a person after a few minutes, 4 or 5 minutes only. That's why we can say that this system is the most important. But we can say it, and because we have very, very high morbidity from of diseases of this system. But definition respiration means much more than only inhalation and exhalation as we understand usually, because respiration have the goal to provide oxygen to the tissues and to remove carbon dioxide, and not only one process, I mean inhalation and exhalation, can provide these many processes. To achieve this, respiration can be divided into 4 major functional events. And these events will be our questions for today. It's external respiration or pulmonary ventilation, which means the inflow and the outflow of air between the atmosphere and the lung alveoli. Next step, next event, is diffusion of oxygen and carbon dioxide between alveoli and the blood. And the third step or event, it's transport of oxygen and carbon dioxide in blood and body fluids to and from the cells. And the fourth event is regulation of ventilation and other facets of respiration. But about regulation we'll talk during next time after New Year. And today we'll consider these steps or these events which provide our tissues by oxygen and remove carbon dioxide. So at first, at the beginning of our lecture, let's remember some knowledge about structural and functional characteristic of respiratory system, which provide external respiration. So Respiration, again I repeat, is a consumption oxygen and excretion carbon dioxide, and includes 4 events: pulmonary ventilation or breathing, gases exchange in lungs, transport of gases by blood, gases exchange in tissues, and tissue respiration. But tissue respiration, it's a subject of biochemistry, and you consider these processes in biochemistry. But first 4 events, it's a subject of physiology. First event, pulmonary ventilation. I told you already that means the inflow and the outflow of air between the atmosphere and lung alveoli. And this event is provided by respiratory system. Functionally, respiratory system consists of 2 parts. You can see here conducting portion, which consists of a series of interconnecting cavities and tubes, both outside and within the lungs. It's nose, pharynx, larynx, trachea, bronchioles, and terminal bronchioles. And function of this portion of respiratory system is filtration, warming, moistening air, and conducting it into the lungs. Other portion of respiratory system is the respiratory portion, which consists of tissues within the lungs where gas exchange occurs. The respiratory bronchioles, you can see here, respiratory bronchioles, alveolar duct, you can see here, alveolar sacs, and separate alveoli in such sacs. And all these structures which are located on one respiratory bronchiole is structural and functional, or structural and functional or respiratory unit, which we call as acinus. Its main sites of gas exchange between air and the blood portion. So, 2 functional parts of respiratory system: the conducting portion, where gas exchange doesn't occur, and the respiratory portion, where gas exchange between air and blood occurs. Here you can see both this functional portion, blue color, it's a conducting portion, and the tissue of lungs, it's rose color, it's respiratory portion of respiratory system. In accordance with these functional portions of respiratory system, we differ to space of air in respiratory system. The volume of the conducting portion in an adult is about approximately 150 milliliters, and this space we call as anatomical dead space, because here we haven't gas exchange. And the respiratory portion much more includes of air. It may be 5 to 6 liters, and its volume is respiratory volume, and this volume we call as physiological dead space, because here we have sorry, because this is my mistake. the volume of respiratory portion unventilated and perfusing is in respiratory portion we call as physiological dead space, because not all acinuses and not all alveoli can be ventilated during our respiration. So we have two types of dead space: anatomical dead space, because anatomically exchange of gases in this portion cannot be, and physiological dead space, because in this case we have alveoli where usually gas exchange occurs, but these alveoli can be unventilated and or can be not perfused, and in this case gas exchange is not occur in this alveoli. That's why we call this space as physiological dead space. Mechanism of pulmonary ventilation, let's consider. Pulmonary ventilation is the mechanical flow of air into and out of the lungs. This movement of air we name into lungs, and outside we name as one cardiac respiratory circle, which consists from two processes, consists of inhalation or inspiration and the exhalation or expiration. And both these processes give us a respiratory circle. It occurs due to pressure difference of the atmosphere and in lungs. And this difference is created by contraction and relaxation of respiratory muscles. So this process is mechanical process, but this process includes one biological mechanism. It's a contraction and relaxation of respiratory muscles that creates pressure difference. Let's consider how this process occurs. Contraction of inhalation muscles provides inhalation, and the muscles which provides this process is external intercostals. You can see here these muscles, and its diaphragm. You can see here line which give us image of position of diaphragm, and its main muscles, inhalation muscles. But when we have deep inhalation, in this case we can observe participation of additional muscles. Additional muscles during labored inhalation, it's scalenus, pectoralis minor, sternocleidomastoid. Actually, it's girdle of superior extremities. Due to contraction of inhalation muscles, we have increase of thoracic cavity and decrease in lungs and in chest of air pressure. And due to these differences, which is created by this contraction, in this case we have increase of the thoracic cavity and decrease of the air pressures in the lungs, and atmospheric air comes into lungs. Да, сразу же. Sorry. Expiration, opposite process, it's a passive process because except forceful breathing, this process occurs in result of relaxation of inhalation muscles and elastic recoil of the lungs, chest wall and abdominal structures, which compress the lungs, and during quiet exhalation. But during forceful breathing, contraction of additional exhalation muscles occurs, and in result of that, we have two groups of exhalation muscles. It's main muscles, internal intercostals, and the additional muscles, abdominal muscles. If you do very strong forceful exhalation, you can feel contraction of these muscles. Due to that, we have decrease of the thoracic cavity and the increase of the air pressure in lungs. And this difference between pressure of air in lungs and atmospheric air, when pressure in lungs more than in atmospheric air, provide movement of air outside from lungs to atmosphere, because we have in this case again difference in different of atmospheric, different of air pressures. Sorry. The distension of the lungs is resisted by surface tension, the thin alveoli, which is kept within manageable limits by the presence of pulmonary surfactant. Surfactant, as you should know already, is a surface active agent which reduces the surface tension, is produced by type 2 alveolar epithelial cells. And it is a complex mixture of several phospholipids, proteins, and ions. Some definitions, some terms which we use in order to describe this event. It's intrapulmonary pressure, it's pressure in lungs, into lungs, and pressure between two pleural layers. You can see here two pleural layers, parietal, external, and visceral, which lie on lungs. Intrapleural pressure, this pressure can be changed during inhalation and exhalation significantly, and due to that, this pressure plays very important role in respiration, external respiration, and in breathing. This pressure in intrapleural pressure is always subatmospheric. So it means that this pressure is lower than atmospheric pressure on 3 millimeters of mercury. Before inhalation, you can see here, because space of this pressure less than during inhalation. And during quiet breathing, we have in inhalation decrease of this pressure on 6 millimeters of mercury, and during forceful breathing, forceful inhalation, this difference can achieve 20 millimeters. It needs say that damage of intrapleural cavity can stop of breathing in spite of contraction of inhalation and exhalation muscles. These muscles can continue contract, but breathing in this case cannot be. Such a situation we have, for example, during such a process as damage of chest. It's so-called pneumothorax, and respiration in this case cannot be realized due to absence of difference between intrapleural and intrapulmonary pressure. In order to estimate this process of respiration, I mean external respiration or ventilation of lungs, we use some methods, and we can describe of this process by some volumes and capacities of lungs. We can divide these volumes and capacities into two groups. It's a static volume and a dynamic volume. Static volume means that this volume of lungs not relate to respiration. It's only volume of pulmonary system. Dynamic volumes give us information about process of respiration. That's why these volumes we call as dynamic volumes. Two static volumes belong next volumes. It's tidal volume. You can see value of this volume, which can achieve a half of liter. Inspiratory reserve volume till 2,000 half liter, milliliter, 2,500 milliliters. It's expiratory reserve volume and vital capacity, which consists from these volumes: tidal, inspiratory, and expiratory reserve volumes. It's residual volume, which can be different in young men and in old persons, and its total lung capacity, which includes vital capacity and residual volume and functional residual capacity, which consists from residual volume, from expiratory reserve volume, and the anatomical dead space. I told you about this volume already earlier. These static volumes and capacities of lungs and dynamic parameters, dynamic volumes, it's respiratory rate, as in heart activity we use heart rate. Here we have respiratory rate, which can be in adults 14-17 cycles per minute. In children you can see more frequently, in newborn again more. It's minute ventilation. I remind you about minute volume in cardiovascular system, which can achieve 6 liters, and which that we can determine by multiplying of respiratory rate on tidal volume during one respiratory circle, volume during one respiratory circle, and alveolar ventilation rate. About this parameter later, I told you. And the methods which we use in order to determine these volumes is spirometry and spirography. Its principle of this method is measurement in first case, when we talk about spirometry, and recording if we talk about spirography of the respiratory air volume. Pneumography, it's method which permit us recording of the movements of the thorax during breathing. And the next method of investigation of this stage of respiration, I mean pulmonary ventilation, it's pneumotachometry. It's the measurement of volume velocity of air stream. And the most effective parameter which can give us information about this stage of breathing is rate of alveolar ventilation. Concerning heart rate activity, concerning heart activity estimation, we use stroke volume in this case, stroke volume and ejection, fraction ejection. Rate of alveolar ventilation is similar fraction ejection, and it's total volume of new air entering the alveoli each minute. And we can estimate, calculate this parameter in accordance with this ratio, where alveolar ventilation is rate, VA, its rate of alveolar ventilation. This is frequency of respiration per minute, and VT tidal volume, and VD it's dead space volume of air. So we can estimate this stage of respiration by such methods as pneumography, spirometry, and pneumotachometry, which give us information about functioning of respiratory system, total functioning of respiratory system, movements of chest during inhalation and exhalation, and mobility of respiratory system and thorax. Methods, spirometry and spirography give us information about different lungs, different volumes of lungs, and so it's information about elasticity of lungs tissue, and about functional state of respiratory portion of respiratory system. And pneumotachometry give us information about functional state of conducting portion of respiratory system, because it's measurement of volume velocity of air stream along this portion, conducting portion. And if we have constriction of this portion and different respiratory ducts in this system, in this case, volume velocity will be more, and we can estimate disorders of functioning of this portion. And these disorders, two types of disorders in respiratory and conducting portion, give us two groups of disorders of respiratory system. It's restrictive disorders, disorders of respiratory portion. For example, in this case, the vital capacity in this type of disorders, vital capacity is reduced below normal, and we can say such situation, for example, at such pathological states and diseases, pulmonary fibrosis. And in our group, when we have disorders of function of conducting portion, it's obstructive disorders when the vital capacity is normal, stays normal, because lung tissues is not damaged. But the conducting portion, first of all, bronchioles, can be constricted, and such bronchoconstriction increases the resistance to airflow, as we can observe at asthma, such disease as asthma. And the increased airway resistance makes expiration more difficult and takes a longer time. So we have just considered first stage of respiration, breathing, or external respiration, or pulmonary ventilation, and we have considered some methods and parameters which give us information about this process. Next stage, it's gas exchange in lungs, and please let's do a break a few minutes before this question. continue. So next step or stage of respiration is gas exchange in lungs. This process means diffusion of oxygen and carbon dioxide between the alveoli and the blood in capillaries, and this process results in, again, difference of gas pressures, and in result of these differences, an increased concentration of oxygen and the decreased concentration of carbon dioxide in the blood leaving the lungs. Independently, pressure of gas in air is called partial pressure. You should know these terms in order to describe this process. And in liquid, blood partial tension. You can see here these differences. In alveolar air, we have approximately partial pressure 100 millimeters of mercury. And in blood, in capillaries of venous and of capillaries, in lungs, we have only 40 millimeters of mercury for carbon dioxide, for oxygen. For carbon dioxide, we have next differences. In alveolar air, we have less partial pressure of carbon dioxide, and in blood, we have 46 partial tension of mercury. Not venous, sorry, I told you here venous. In arterial end of capillaries here 40 millimeters of mercury. So we have such gradients for oxygen and for carbon dioxide. It's 60, it's easy, remember, 60 millimeters for oxygen and only 6 millimeters for carbon dioxide. And these differences create conditions in order to gas exchange in lungs and diffusion through lungs tissue to capillaries can be. This gas exchange occurs through respiratory membrane, and this membrane we call as hematoalveolar barrier. That consists of next layers. It's layer of surfactant, you can see here, blue color. Surfactant reduces the surface tension of the alveolar fluid. It's alveolar epithelium, which is composed of thin epithelial cells. It's epithelial basal membrane. It's a thin interstitial fluid between the alveoli epithelium and the capillary membrane. It's a capillary basal membrane that in many places fuses the epithelial basal membrane. And it's a capillary endothelial membrane. So oxygen and carbon dioxide, in order to be exchanged, should pass through all these layers. And the structural and functional state of this membrane, this barrier, determine this process, second stage or process of gas exchange in lungs. This process depends on and describes by diffusion coefficient, which depends on gas solubility in the membrane, and inversely proportion on square root of its molecular weight. And the ability of the respiratory membrane to exchange a gas between the alveoli and the pulmonary blood can be expressed in ratio, in quantitative terms, by its diffusion capacity. What does it mean? It's volume of gas that diffuses through the membrane each minute for a pressure difference of 1 millimeter of mercury. Its diffusion capacity. So we can measure diffusion capacity as the volume of gas that diffuses through the membrane each minute for a pressure difference of 1 millimeter of mercury, and this capacity depends on relation alveolar ventilation divided perfusion, or related to perfusion. Perfusion is blood flow in lungs, and this relation you can see here. It's blood flow and it's alveolar ventilation, and effectivity of this stage of respiration is determined by this relation, ventilation to perfusion, alveolar ventilation to perfusion. We know that upper portions of lungs are ventilated less, so efficiency of gas exchange in lungs depend on this ratio, and method by which we can estimate this next stage of respiration, gas exchange in lungs, is analysis of gases content in alveolar air. You can see here on the slide composition of different airs. Atmospheric air, it's inspired air or alveolar air, where oxygen is approximately 21% of total volume of gases, carbon dioxide only 0.03%, and the most part is nitrogen, approximately 8%. Expired volume, expired air consists less quantity of oxygen, more carbon dioxide, and almost the same quantity of nitrogen. And alveolar air, expiratory alveolar air during exhalation, includes less oxygen, more carbon dioxide, and a few more nitrogen. Why we have such differences? Atmospheric air, it's clear, it's alveolar, inspired alveolar air. It's composition of our atmospheric air, our atmosphere. Expired and alveolar air have some differences in quantity of gases, because expired air, it's a mixture between air of anatomical dead space, where gas exchange is not does not occur, and alveolar air, it's a composition of air which fully will be, not will be, would be exchanged in alveoli. So we have on this picture exchange between gases during a second stage, or second event of respiration. It's gas exchange in lungs, where oxygen from alveoli comes to blood, and carbon dioxide from blood, venous blood of lungs, comes to alveoli and will be exhalated. This expired during exhalation. And I remind you that this process is possible due to pressurous gradient between partial pressure for each gas in air and partial tension for each gas in blood as liquid. We will not come back to description of process of gas exchange in tissues in order to economy our time. That's why I told you here that we have the same laws of gas exchange in tissues, because here movements of gases, oxygen from capillaries to interstitial fluid and to cells, and carbon dioxide from cells to capillaries is possible due to pressure's gradient between partial tension of gases in blood and partial tension of gases in interstitial fluid and in cells. And these pressures differences you can see here. For oxygen, it's 40 millimeters of mercury in bloods, and for carbon dioxide, 45 millimeters, and in cells, partial tension for oxygen achieved 0, because oxygen is included in metabolism in cells, and we cannot almost free oxygen in cells. That's why partial tension is 0, and vice versa. Carbon dioxide in cells, partial tension for carbon dioxide in cells achieves 60 millimeters of mercury. That's why we have movement of oxygen to cells, intercell cells, and carbon dioxide in opposite direction. Third stage, it's transport of gases. After gas exchange in lungs, oxygen comes to into blood, into capillaries, and transportation of oxygen is realizes by in combination with hemoglobin. Hemoglobin maybe you know, not maybe, you should know already that hemoglobin is included in its most structure, not structure, molecule substance of in erythrocytes, and oxygen principally is transported by in combination this hemoglobin. Such association we name as oxygen hemoglobin or oxyhemoglobin. And in lungs, in capillaries of lungs, oxyhemoglobin is quantity of oxyhemoglobin is 98%. Only 2% of hemoglobin is not associated with oxygen. And the last part of oxygen can be dissolved in plasma and transported in such dissolved states. Concerning carbon dioxide, we have more forms of transportation, and the most part you can see here, it's 70%. 70% of carbon dioxide is transported in a form of bicarbonate ion, and a very important role in creation of this form bicarbonate salts. Very important role in creation of these substances play carbonic anhydrase, such enzyme. Besides this, carbon dioxide can be transported in combination with hemoglobin and plasma proteins. It's carbaminohemoglobin or hemoglobin carbon dioxide, which is approximately 23%, and approximately 7% of carbon dioxide can be dissolved, can be transported in dissolved state. You can see here transportation of oxygen in combination this hemoglobin in the dissolved state, oxygen from lungs. This forms of transportation of oxygen comes to tissues, where oxygen comes to cells, and carbon dioxide, vice versa, comes from cells to blood, and where carbon dioxide can create bicarbonates or associates this hemoglobin, and the last part dissolved part, and in such forms of transportation, carbon dioxide comes to alveoli and go out from organism. This process, creation, formation of all these combinations, all these forms of transportation can be described by a curve of dissociation for oxygen and for carbon dioxide. Usually we consider and we use estimation of this transportation for oxygen. curve of which describes dependence between partial pressure of oxygen in lungs and the percent of hemoglobin, oxyhemoglobin, percent oxyhemoglobin saturation. This curve we name as a curve of oxygen hemoglobin dissociation curve. This curve describes processes which occur in lungs. It's such part of this curve where we have the highest partial pressure of oxygen and partial tension of oxygen in arteries, approximately 100 millimeters of mercury for oxygen. And this part of this curve, it's processes which occurs in tissues where partial tension of oxygen is 0, and due to that we have in lungs the highest percent, 98%, I remind you, of oxyhemoglobin, creation of oxyhemoglobin, and the dissociation of this complex during movement, during transportation of oxygen to tissues where partial tension of oxygen decreases, and the association of this complex occurs easy due to that. But not only partial pressure or tension for oxygen can determine this process. On this process can next factors influence. It's factors affecting hemoglobin affinity for oxygen is acidity, pH, concentration of hydrogen ions, temperature, and partial pressure of carbon dioxide, and such chemical substance which is which presence in erythrocytes and influence on dissociation of this complex oxyhemoglobin. All these curves demonstrates a progressive increment of the percentage of hemoglobin that is bound this oxygen as the blood pressure carbon oxygen increases, as the blood P oxygen increases, which is called the percent saturation of the hemoglobin. And we can observe that these curves can be moved in right or in left, depending on values of these factors. For example, here you can see changes of pH. Low pH and low acidity, and this low blood pH you can see here, and more, acidity in this case. In this case, we have how process of dissociation of oxyhemoglobin and movement of oxygen to tissues can be changed. Such position of curve we call as right shift of dissociation. And in this case, on the left shift, you can see here. Dissociation of the oxygen hemoglobin occurs more easily in conditions when the temperature higher and pH is lower. And you can see here shifts to right and shift to left at the changes of temperature and carbon dioxide pressure potential. So these factors can influence on process both combination or association of between oxygen and the hemoglobin and dissociation of this complex. That's why in some cases when we have enough during breathing, enough quantity of oxygen in blood, not we can have a situation when dissociation of this complex is difficult and the tissue cannot get enough quantity of oxygen. For example, when patient is illness, and in this case, not enough quantity of oxygen, tissues of this patient cannot receive. Some terms which we use in order to describe this process, it's oxygen blood capacity, it's volume of oxygen that can be connected by 1 liter of blood. In order to determine this capacity, we need 1 molecule of hemoglobin, which can get connected 4 molecule of oxygen. Calculate that 1 gram, if we know that 1 gram hemoglobin can get connected 1 milliliters, 1 milliliters 34 1.34 milliliters of oxygen. It's so-called number of Hefner, and taking into account these quantities, we can estimate that 1 liter of blood can get connected 0.2 liter oxygen, or 5 liters of blood can contain 1 liter of oxygen. So oxygen blood capacity is 1 liter on 5 liter of blood. 1 liter of oxygen on 5 liter of blood. And over terms, parameter which we use in order to describe this process, this stage of transportation of gases, it's oxygen utilization coefficient. Different tissues can different abilities to utilize the oxygen, and this coefficient is portion of oxygen that is saturated by tissues. The highest ability to saturate of oxygen is we can meet in heart. Actually, this coefficient more in tissues which we have higher intensity of metabolism, because in this case more oxygen is needed to these tissues. But I repeat again, the highest utilization coefficient, oxygen utilization coefficient, we have in heart. Okay, that almost all about different stages, 4 main stages of respiration. I remind you that this respiration is much more larger definition as only breathing, because respiration it's consumption of oxygen and remove carbon dioxide, and it is sum of different processes which provide gas exchange in lungs, gas exchange between atmosphere, between external environment and lungs, between lungs and blood, capillaries in lungs. It's processes of transportation of oxygen and carbon dioxide, first to tissues, and carbon dioxide from tissues to lungs, and it's a process of gas exchange in tissues. And all these processes influence in final event, it's a consumption of oxygen in tissues. But usually in clinical practice, we use mainly investigation of breathing, because I repeat again, diseases of respiratory system plays in leader have leader places in morbidity of population in whole world. That's why we first of all should know how respiratory system functions, but we should take into account that respiration includes not only that process. Very important role plays both over events of respiration, it's gas exchange in lungs, and transportation of gases. Gas exchange in lungs depend on functional state of hematoalveoli barrier, and in this case we should that we have in atmosphere now very many pollutants which can damage of this process, gas exchange in lungs, and who are smoking, for example, who smoke can damage this barrier as well. Okay, it's almost all concerning respiration, and today we'll start consider next topic of this next question of this topic. It's regulation of respiration. I'll start to consider this material and will continue during next lecture. First of all, it needs to say that regulation of respiration has, if I can say so, the main aim as goal is supply the needs in oxygen of our tissues. And this regulation aimed to changes of quantity of oxygen in blood, which will be delivered to tissues, independent on functional state. If we are in a relaxed state, of course, this needs is lower than during physical loads, intensive physical loads. And in order to achieve adequate supply by oxygen our tissues, our organism use different such means as frequency, changes frequency, and the depth of the breathing. We observe the same situation in regulation of the heart activity, for example. You have seen already that. And in respiratory system, we have very, very important peculiarity because level of regulation here only central. Why only central? Because mechanism of respiration, it's mechanical process which is provided by contraction of respiratory muscles. Respiratory muscles are the skeletal muscles, and skeletal muscles are innervated by somatic nerves. And somatic nerves, as you know, can be regulated consciously. That's why we can regulate our breathing, and in this case we have only central regulation. We haven't humoral regulation of respiration because we haven't direct influence of humoral factors on contraction of respiratory muscles. But in some manuals you can meet such definition as humoral regulation of respiration, but in this case we can consider it is not exactly correct, and I'll explain you why later during this lecture, during next lecture. If we have here only central regulation, central level regulation of respiratory system, it means that in regulation of respiration we should consider reflex arc of respiratory reflexes, and this reflex arc consists, it's usual, from efferent, central, and efferent limbs. Each from this limb we will consider during next lecture. So today we have considered how breathing occurs, what events in supply by oxygen of our tissue we have besides breathing more, gas exchange in lungs, gas exchange, transportation of gases and gas exchange in tissues, At next time, we'll consider how regulation of respiration occurs. First of all, we mean in this case regulation of first stage of respiration, regulation of breathing. Maybe that's all for today, my dear friends. If you have some questions, you can ask me in chat. Please, I'm waiting for your questions. My dear friend, we haven't meetings during last lectures, and I don't understand why number of students during this time so much decreases. Nobody want to ask me. Okay, I'll ask you during exam. Okay, my dear friends, I wish you good luck in preparing of physiology, and goodbye.